Hello everyone, my name is David Twesigomwe. I'm from the Sydney Brenner Institute for Molecular Bioscience at the University of the Witwatersrand in Johannesburg. I'm presenting a poster from my master's work titled Characterization of CYP2d6 Pharmacogenomic Variation in African Populations, an Integrative Bioinformatics Approach. Pharmacogenomics is the study of how genetic variation influences drug response. CYP2d6 is one of the most important pharmacogenes as it metabolizes about 25% of commonly prescribed medications. However, genotyping CYP2d6 is quite challenging due to the structure variants in the gene locus. I've included this image from Twist et al. 2016, and it shows a number of haplotypes in CYP2d6, or star alleles as we normally call them. So here we see CYP2d6 located on the minor strand of chromosome 22 alongside two pseudogenes, CYP2d7 and CYP2d8. There is a 94% homology between CYP2d6 and CYP2d7, and this is responsible for structural variants such as these hybrid genes in part D of the figure and tandem rearrangements in part E of the figure. We also have other variants such as the gene deletion, and which is called star5, and the gene duplication. So these variants really make sequencing this region difficult, and also alignment of short reads really difficult. Another thing to note here is the functional annotation of these haplotypes. So we see the normal function alleles, decreased function alleles, and non-functional alleles. So our objectives were to compare the performance of CYP2d6 star allele coding algorithms, and then to identify CYP2d6 star alleles in African populations using whole genome sequence data, and then to predict the CYP2d6 metabolizer phenotype distribution in African populations, and also determine the frequency of the regulatory variants in African populations. So from the workflow, we can see the two parts of our study. The first part is a benchmarking study where we use 75 reference datasets from the genetic testing reference materials program. And here, the tools we compared are ALDI, Astrolabe, and Stargazer. We also compared whether using an ensemble genotyping approach involving all the three tools is better than using only one tool. And afterwards, we used that consensus genotyping approach and applied it to 458 African whole genome sequence samples. And these were mainly from Southern Africa and Western Africa. And they were originally from the H3 Africa Consortium. And also we had some samples from the Simon's Genome Diversity Project. After calling star alleles from the African data, we used the activity score system for the phenotype prediction. And here I've included on the right a breakdown of how we score different alleles based on their function. From the results, we see that ALDI and Stargazer perform much better than Astrolabe in calling CYP2d6 star alleles, and this is because they are better at calling alleles in samples with CYP2d6 structure variants. We also see that using the ensemble genotyping approach is much better than using either tool alone. So this is the distribution of the star alleles in our data set. So our data set is represented by the HAD in red here, and this is the high coverage African ADME data set, and we compare that to other previous studies in sub-Saharan Africa and other global populations. So here I've highlighted the gene deletion, which is star5, and we see that there's a much higher frequency of star5 in African populations compared to other populations. And then we also see star 17 and star 29 being really African specific, but they also have a high frequency in admixed Americans. These two alleles are decreased function alleles, and they are responsible for a lot of the enzyme variability that we see in African populations. And then also here we see the structural variants, which we were not going to be able to call if we had not used whole genome sequence data and the tools that we used. Then here we see the phenotype prediction. We see that there are more ultra-rapid metabolizers in Western Africa compared to Southern Africa. And then we also see that there are more poor metabolizers in Southern Africa compared to Western Africa. And this is due to the different distribution of the star alleles in the different populations. We also see a contribution by the long-range regulatory polymorphisms, and these two, for example, are in linkage disequilibrium, and they are located about 100,000 base pairs away from the CYP2d6 gene. So in conclusion, combining whole genome sequence data and an ensemble of star allele calling algorithms facilitates accurate genotyping of CYP2d6. Then the CYP2d6 activity score facilitates prediction of the metabolizer phenotype from genotype. However, other factors such as long-range enhancer polymorphisms may play a role in regulating CYP2d6 expression. With that, I would love to thank the following: my supervisors, Prof. Scott Hazelhurst and Professor Neil Lombard, the ADME project team here at WITS, and our external collaborators, GSK for funding, and as well as the NRF, and the International Society for Computational Biology for the ISMB 2020 Fellowship. Thank you.